So is it visible now? Hello, hello.
Yeah, it is clearly visible.
Okay, 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 okay. Yeah, I'm just checking because I'm not
Yeah.
used to with this Google Meet. Yeah, so
Okay,
we
no
have been
problem. Actually,
using
it is
the Zoom. Okay, I think it should be fine. I think.
yeah, it's clearly visible.
So is this entire slide visible or like or only partial one? Can you see
No,
the
no,
entire slide?
entire slide. Yeah, entire slide is visible. Some text is coming, and besides that, one picture, two pictures are.
This
The whole interface is coming.
and
Uh,
even the cursor is it, isn't it?
Cursor is also moving.
Okay, okay, okay. I think it should be fine.
Students are also joining. Actually, today we have our conference, so that's why it is they will join there.
Okay.
I think
Yeah, yeah.
before 4:10 or 4:05, all will join.
I see. Okay. I think it's morning 6 a.m. here. I think
Yeah,
5:30.
yeah. So you can understand. Actually, very sorry for that. Actually, students are doing their classes from 8 to 3 or 8 to 4. Actually, so that is the problem. Before 4, it is not possible.
So
So
I think
that's why we are trying to. Yeah,
most of the students are undergraduate, isn't it?
yeah, undergraduate students.
Okay, okay. So they are like which year? I think probably second year,
They're actually third year and fourth year students.
third year, and fourth years. So I think they might be knowing much better than me. I think I'm not a
No,
computational guy.
actually, that is no. Actually, machine learning they have been not taught in detail. So that's why we thought if you tell about some machine learning in some different area. Obviously, they are from computer science background, but actually they didn't or they don't know actually how the machine learning can be helpful in other areas also. So if
Okay,
this talk will help them, then they can use this machine learning techniques in other areas also. They are
I
thinking
see.
that always this machine learning belongs to computer science.
Okay, okay, okay. So I am just using it as an application, yeah, in biology, biomedical.
So generally they become free from 4 to 5. So that's why we have planned to keep this time slot from 4 to 5.
Okay, okay. Yeah, I think it should be no problem.
So please go to the second slide. Is it coming properly or you have to again manage it? The next
Okay.
slide, is it coming?
Let me check. Is it fine?
It is coming. It's fine. It's coming. Yeah.
Okay.
Yeah, it's coming properly. Yeah. So we have to wait for another 3 to 4 minutes. Students are
Yeah,
actually joining.
it's fine.
Ma'am, Vishnupriya ma'am,
Yes,
good
yes,
evening.
ma'am. Yes, Madhuri
Uh,
ma'am.
Good evening, Abhimanyu sir. Thank you for joining.
Actually, your
Actually, we
voice
had
is
about,
very low, ma'am.
ma'am, we had about 134 registration students are slowly joining. I think it's already 4 5. Let us start. I think more will join because we had the conference and again,
Yeah, yeah.
but we have
So
134 registrations, so people will join.
we'll wait or we'll start, ma'am.
I think by 4 5, it's 5 minutes. So let us start.
Okay, let's start. Okay.
Students will join like this.
Okay,
Okay,
okay.
then.
Since we have actually the conference, that's actually above 130 students they have joined actually. So due to that
Okay.
problem, conference problems, if you okay, so let's start. So, good afternoon, all participants, those who have joined here. Um, yeah, one another person is coming. So, good afternoon again, all participants. I, Dr. Vishnupriya Panda, coordinator of 11th lecture of weekly webinar series of SOA. Welcome you all. I also welcome the convener of webinar, Professor Devoti Mishra, ma'am, and all faculty organizers like Dr. Sarada Prasad Pati sir, Dr. Bharat Jyotiranjan Sao sir, and Dr. Deepasundar Das sir, and Dr. Madhuri Rao ma'am. It's my pleasure to welcome today's speaker, Dr. Abhimanyu Thakur. And Dr. Abhimanyu Thakur obtained PhD in biomedical sciences at the City University of Hong Kong by investigating the role of glioma-derived exosomal surface proteins as biomarkers and functional mediators. Next, he joined as postdoctoral fellow at Hong Kong Institute of Science and Innovation, followed by in you can Briscoe School of Molecular Engineering, then made Department of Cancer Research at the University of Chicago, Illinois, USA. His postdoc research is focused on the crosstalk of stem cells-derived exosomes in health and disease. Besides, he has interest in the interdisciplinary biomedical sciences, including biomedical application of machine learning. His researches have been published in reputed peer-reviewed journals, including Science Advances, Biosensors and Bioelectronics, Advanced Functional Materials, and Chemical Engineering Journal. His current Google Scholar citation is more than 600 with H index 11. He has been conferred with various prestigious awards such as International Research Abstract Award in 2020 by Korean Cancer Association, South Korea, Young Investigator Award in 2019 conferred by American Association for Cancer Research, Service Appreciation Award by Hong Kong Precision Oncology Society in 2018, then CTU Outstanding Academic Performance Award in 2018. He also received prestigious Global Young Scientist Summit 2018, organized by Nanyang Technological University, Singapore. And it is my privilege that we are also working together and have published previously. So now I welcome Dr. Thakur to deliver his talk. So Dr. Thakur, it's over to you. So please, you can continue.
Yeah. Yeah, thanks, Dr. Panda, for this wonderful opportunity, and I'm really privileged to have this opportunity. And also thanks to Madhuri Rao, like, yeah, for greeting
Yeah,
me.
Madhuri.
Yeah. So,
Thank you, sir.
I think, yeah, I should start. I think. Yes. So, hi friends. As you know, like, my name is Abhimanyu Thakur, and so I am a biomedical researcher, and I work generally on cancers, like, in particular brain cancer. It's also technically you call it as a glioma. So among several different research projects, I have been also working for several years on machine learning in cancer diagnosis and research. So in today's talk, I will be giving a brief about like how we can apply this machine learning in different aspects of cancer research in terms of diagnosis as well as biomedical research. Is it visible now? Hello.
Yes, yes, it is clearly
Yeah.
visible.
So, friends, like, there are several diseases, like, and those are like fatal diseases like cancer, and even like progressive diseases like Alzheimer's, like Parkinson disease, and all such disease like needs attention, as the survival rate is very low and the cost of care is very high. And by the time we detect it, the disease might have already surpassed the advanced stage. So therefore, there is a demand for the early diagnosis of this kind of the fatal and progressive diseases, because an early diagnosis will, like, it will open a door to the future care and treatment. So here I would like to take a specific example of cancer, that is glioma, which is also known as brain cancer. So this one will depict why the early diagnosis is crucial. For example, glioma, it's a brain cancer originating from mainly neuroglia stem cells in the central nervous system. And according to the latest statistics, around 1.71 million patients are affected by glioma. And this is one of the most aggressive and malignant form of glioma is the glioblastoma. And importantly, even though there are combination of treatments available for this glioma, including like temozolomide, radiation therapy, and surgery, but still there is a but still the average survival rate is around only 15 months. And so uh, you can understand a person suffering from a disease like brain cancer is already on deathbed, and therefore it's very important to develop a diagnostic method for early diagnosis right from the beginning of any molecular event before it leads to any particular like clinical symptoms. So that shows how crucial it is to have a method which can diagnose this kind of fatal disease right before if it has occurred. So there is in next example, I will let you understand about how this can be used even in the neurodegenerative diseases. So you might have seen that you might have heard about Stephen Hawkins or even Superman. So these guys have been suffering from a progressive neurodegenerative disease like amyotrophic lateral sclerosis. What happens is this is also one of the neurodegenerative disease which is a progressive one and involves like slow degradation of neurons, include leading to various clinical symptoms and including dementia. So they lose their uh, ability or power to remember anything. So the dementia, they suffer from dementia. And the two important molecular events which is crucial for development of Alzheimer's disease, including like decreased levels of amyloid beta. So this is one of the protein which is decreased in the Alzheimer's disease, and increased level of phosphorylated tau in cerebrospinal fluid. So the available methods for the diagnosis including either like invasive or they are very costly, which which again creates a huge burden on the patients and their family. Therefore, again there is a demand for developing non-invasive or minimal invasive biofluids based diagnostic method, which should be like robust and can be easily handled. So that shows the need for that shows the need for like some non-invasive method which can detect these kind of fatal diseases like very early. So as we can see, there are several imaging devices like computed tomography, CT scan, or MRI, MRI, which are although gold standard but suffer various limitations such as exposure of patients to harmful radiation or injecting contrasting dyes in the patient. And moreover, the tissue biopsy is highly invasive and requires professional handling. Most importantly, there are painful and incurs discomfort in patients. So here comes this liquid biopsy in the picture. There are several biofluids, analytes, and biomarkers encompassed in liquid biopsy. For example, in 2013, US FDA approved first liquid biopsy test, that is cell research, and uh, it it intend to monitor advanced metastatic breast cancer, colon, or even prostate cancer based on CTC counts. And 3 years later, in 2016, like the first ctDNA based liquid biopsy test was approved. A comparison test to detect EGFR, that is one of the mutations in the ctDNA of patients with non-small cell lung cancer, which may benefit from the targeted therapy with the well-known popular drugs, that is erlotinib and osimertinib. So basically my point is this kind of liquid biopsy is already in the market, like right from 2013 it has already been approved, and also in 2016, as you can see. So let's hear it. What are these biofluids in non-invasive diagnosis? So most recently, advances in the detection and characterization of this ctDNA have finally enabled the introduction of liquid biopsy assays into clinical practices, and the FDA has already approved cell-free DNA, as we saw in our previous slide, and multi-gene assays to detect genetic alterations in plasma cell-free DNA for using as comparison diagnostic match to specific molecular target therapies for cancer. Well, I think my talk might sound like too much technical. So let me know if you don't understand any kind of terms. So in simple word, these cell-free DNAs are like always in our blood, and this can be tested using this non-invasive liquid biopsy method. So what I'm saying is, whenever there is a tumor growth, so I'll explain it a little bit in simple way. So whenever there is a tumor growth, so there are certain DNA or even nucleic acids, they get ooze out, and they pass it into the our blood, and that can be detected. So they are basically constantly moving in our blood. And right from even before its onset, there are probability of these ctDNAs, they can be found even in our blood. That's why a single blood sample can contain a huge range of cell types or even cell products, like emanating from multiple tumor sites around the body. And these liquid biopsy assays of these diseases, cells derived factors can serve different purposes in their management. For example, it can be used to further investigate the abnormalities, like, and they can detect on imaging examinations such as mammography or lung CT, or it can also be used for surveillance for the disease following the treatment, or even the doctor can prescribe it like after the like post treatment surveillance, or even it can be used for guiding the selection of the most appropriate treatment or the combination, or or for monitoring treatment response in patients through dynamic characterization of changes in the disease biology. So one of the crucial player in liquid biopsy is the exosome. These exosomes are tiny vesicles with size around 30 to 200 nanometer, which are released by most cells biofluids, including blood and cerebrospinal fluid. And these exosomes contain tumor-specific mRNA and mutant proteins, including EGFRV3. So this is one of the mutant protein which is present in this kind of exosome, and it has been found that these tiny extracellular vesicles, they can cross the blood-brain barrier or even the blood cerebrospinal fluid. Uh, so these are the important barriers which separates our brain from other parts of body. And interestingly, these exosomes can cross these important physiological barriers in the brain, and they can or they can mimic their parent cells from which they have been derived, and that's why they can mirror their parent cells. So it's a good platform for the biosensing of biomarker to track uh, like various brain diseases, including glioma. So, well, recently last year, our work on exosomes-based liquid biopsy, our lab group, we published a science paper, and it got featured in this Times Higher Education group, and it was also shown up in various news agencies like Asia Research News. And yeah, so in our work, we utilize the concept of tumor metabolism. In 1931, Otto Warburg, he got Nobel Prize as he demonstrated that all forms of cancer possess acidic environment as well as hypoxic, that is less amount of oxygen. So similarly, in our work, we proposed how this metabolic reprogramming is important for the survival of cancer cells. One of the key hallmarks in most of the malignant progression of the cancer, or including glioma, is closely associated with the metabolic reprogramming towards aerobic glycolysis, which is known as the Warburg effect. Therefore, the malignant glioma cells, these increase the glucose consumption and lactate production through rapid glycolysis to meet high demand of energy for their energy growth and migration. Importantly, the malignant glioma cells, this enhance the two important glycolytic proteins, that is MCT1 and CD147, and the expression to pump this intracellular lactate out of the cells for the maintenance of continuous glycolysis. This is an important step for the energy in our body, resulting in the accumulation of lactates in the tumor microenvironments. So you might have heard about glycolysis, Krebs cycle, like glycolysis occurring in the cytosol of the cell, and Krebs cycle occurring in the mitochondria. So these are important physiological steps which takes place in our body system for building up energy. So here whenever there is a cancer, so there is a less oxygen, there is a occurrence of hypoxic condition, which is basically less oxygen in the tumor site, and then there is increased lactate inside this. Yeah, can you see my cursor? Hello.
Yes, 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 yes.
Yes, sir.
Your question is
Yeah. So yeah, so suppose this is a tumor, and then there is an increased lactate inside the tumor, and then this lactate is not good for this tumor site, so it has to go outside. So that's how these two important proteins comes into picture, and they get overexpressed, and they move out this lactate out of the cells, and then there is an increased extracellular lactate, and they form a metabolic coupling, and together they help in this tumor progression. So these extracellular lactates also can be taken up into the surrounding fasting glioma cells and other stromal cells like neighboring cells in the hypoxic tumor microenvironment, and establish a beneficial metabolic coupling among the heterogeneous neighboring cells. So this MCT1, it's a major monocarboxylate transporter in the central nervous system, which play a crucial role in transport of the lactate across the cell membrane by cooperative action with its binding partner, that is CD147. And this MCT1 and CD147, they together in the various tumor cells, including glioma, they are significantly upregulated during the malignant progression. Therefore, the level of distribution in glioma tissues have been considered as a crucial indicator to determine the glioma malignancy associated with metabolic adaptation. And therefore it is like we can thought that this, by blocking this the function of MCT1 and CD147, can suppress the growth, metastasis, and invasion of the glioma cells, as well as angiogenesis, leading to the development of the inhibitors as anti-glioma agents. So the hypoxic tumor microenvironment, so the hypoxic uh, tumor microenvironment, the expression of MCT1 and CD147 is increased. In addition, the malignant GMS release a significant high number of exosomes. But we wonder how the increased expression of MCT1, CD147, they control the enhanced release of exosomes from glioma. So, well, the available standard methods for the diagnosis of glioma, like MRI or even CT scan, but these are challenged for the detection of precise molecular signature. Uh, excuse me. Yeah, precise molecular signature, precise precise well these method, there are many methods like MRI or even CT scan, and there are many biochemical methods like electron microscope or ELISA or western blot. So these are always used in the laboratory settings, but these are time consuming, or even they are indirect method, or they have limited sensitivity, or there is a chance for the protein contamination, or even there is a requirement for the large volume of sample. So the that's how these are challenged for the detection of precise molecular signature of glioma progression, which demands again a non-invasive biomarker for the precise monitoring of malignant glioma progression. Therefore, we have utilized LSPR, that is localized surface plasmon resonance, and AFM, atomic force microscopy based biosensors for the detection of exosomal biomarker. So what is this LSPR? It's a label-free technique with high sensitivity, which can be used for the detection of various interactions such as antigen-antibody, protein-protein interaction. And here is a representative setup. This is a representative setup of LSPR and SPR sensogram. This is a SPR sensogram. Uh, and in our study, we have utilized the special engineering based chip that is self-assembled monolayer gold nano island LSPR. So it's one of the bioengineering tool which we utilize for the detection of those exosomal proteins. And in another method, that is AFM, atomic force microscopy, which is again a high resolution scanning probe microscopy with a resolution of fractions of a nanometer. And here is a representative setup for the AFM. This cantilever is used for the detection of the sample, and it has a AFM tip. This is a AFM tip, and these are the AFM force curve, typical AFM force curve. Traditionally, researchers have used like uh, like mica, or even the gold, over the mica or the glass surface, for capturing and detecting these exosomes. And in our study, again, we used gold nano island sample discs for better resolution. So here is the experimental data about the exosomes biosensing by the SPR and AFM. So which we use for the non-invasive sensitive detection of the increased MCT1. So here you can see, like in the hypoxic condition, there is an increased level of MCT1 and CD147 both. So you can see there is a significant increase. And exosome, and again in the AFM also we found the similar pattern. So this is the typical 3D diagram for the exosomes, and this is the two-dimensional diagram. And here you can see the height curve for these exosomes, uh, this exosomal structure, nanostructure. So what have we discovered? Yeah. So what we have found is hypoxia in tumor microenvironment. They enhance the lactate, and for its exocytosis, MCT1 and CD147 play a crucial role by migrating to the cell membrane, and there is an calcium, enhanced level of calcium, which facilitates the release of exosomes, which can be taken up by the neighboring these endothelial cells, and lead to the tube formation, and an event in the angiogenesis, which is again crucial for the like third stage of or fourth or the final stage of brain cancer. As mentioned earlier, exosomes, they impersonate their parent cells. Their constituents were detected by LSPR and AFM biosensors. So in one of the we used one of the engineering tool, and we published our this detection system in advanced functional material. And this one we used it for the detection of this EGFRV3. This is one of the mutant protein in the most of the uh, EGFR is specifically in the glioma cells, and here we found in especially glioma cells derived exosomes. And here you can clearly see that in the kidney cancer cells derived exosomes, there is a very few EGFRV3 mutant protein, and which we again detected using this LSPR tool. So considering all these, here we can see, although there are various methods like electron microscope, dynamic scattering technology, screening technology, or even nanotracking analysis, these are available for the characterization of exosomes, but all comes with certain limitations. So so we need to overcome this one. So so far I have been explaining why we need this machine learning. So a lot of talk about this biology. So I know it might have surpassed the brain of most of the computer engineers. So like let's know how we can overcome, how we can overcome this problem. So here I made a plan. This we might have like certain scope by integrating the machine learning and this exosome biology, and we can use like neural networks that can be stunningly accurate at making predictions. So in as a first example, as one of the example I'll talk about like how the for the detection of disease specific parent cells via distinct population of nanovesicles by machine learning. So as mentioned earlier, these exosomes are released from any kind of cell, including the dendritic cell, macrophages, lymphocytes, and tumor cells through exocytosis to the extracellular space, and always found in any human body fluid such as saliva, urine, plasma, breast milk, or ascites, as well as the blood. These cells derived particles contain nanoparticles contain microRNA, mRNA, glycans, proteins, many kind of biomolecules. These are specific to most of the host cell types, and have a lipid membrane whose composition retain characteristics of the host cell membrane. So here I took one example. I'm taking one example of age-related muscular degeneration, which is related with the eyes, and using this angiogenesis phenomena in the cancer microenvironment by using these endothelial cells. So what we did is we isolated these extracellular vesicles like exosomes and other kind of vesicles. There are like microvesicles, and together these are all like the nanovesicles we termed it as. And then uh, I we measured its size distribution, and recorded various parameters, including like surface density, and there are other kind of attributes which we took into consideration. Then what we did is we used this data set and divided into two different subset, that is training data set and test data set, in the ratio of like 80% and 20%. So and then we used various kind of machine learning models like quadratic support vector machine, or cubic support vector machine, Gaussian support vector machine, fine tree, and back tree. So these are all like decision tree models. So we trained these models on our training data set, and we asked the question whether this can be used for predicting what kind of cancer cells, or what kind of cells rather to say, from which these vesicles have been derived from. And then we measured the performance. So let's see the results. So these are the size distribution pattern for these two different nanovesicle. One is this derived from the RP, retinal pigment epithelial cells, and another is from the endothelial cells. So here we can clearly see like how the endothelial cells, they are relatively less compared to these RP derived nanovesicles. And even with the different size parameters, they have like consistently they've been increasing. Like most of the vesicles are around D90, that is their diameter is around that is partially around 400 nanometer. Like 90% of this diameter are around 300 to 400 nanometer. So this is the performance. This is we prepared ROC curve for measuring the performance. And here again we can see, out of these several different models, we found the maximum in the decision tree, and among the decision tree we found maximum in the this back tree model. You can clearly see like how there is a maximum area under curve showing up. Then we analyzed our data, and we found, and to our surprise, we found that in the back tree there is a highest prediction rate compared to other this model systems, machine learning based model systems. So what we found is by the use of this machine learning, there is a dramatic decrease in time consumption. So if we make a comparison based on the time, so for example, sample preparation in the previous method, depending on the technique, again, so this is almost same. Sample processing time, it generally takes 1 day, but in our this AI based methods, we don't need any kind of processing, so it saves our 1 day. And then sample analysis, it takes again 1 to 3 hours, like immobilization on the substrate and consequently deformed. So this can be correlated with the data which I showed up, which I did, we did, which our lab group did in terms of biological experiments. So here in the AI based models, only taking 15 minutes. So again, it is saving 45 minutes around. And in the data processing, like image processing, dimensionality reduction, all these are required. But in AI based method, this is like ready to use. So saves us again significant amount of time. So conclusion, these exosomes are an important tool for non-invasive liquid biopsy, and the application of the machine learning algorithm on the features of exosomes they derived from biofluids. This can be an important tool for the early diagnosis of many different fatal diseases, including cancer. Particularly, I talked about glioma, that is brain cancer, and with this we can be used for the development of potential diagnostic methodology to track progression of fatal diseases. Yeah, thanks for listening. So feel free to ask and collaborate. This is my email ID. If you have any question, you can email me in future. Yeah, thanks.
Yeah, thank you, Dr. Abhimanyu. So here I would like to ask our participants, do you have any questions? You can directly ask to him, otherwise you can write on the chat box. And also he has given his mail ID, and if in future you want to, contact with him, you can contact with him and you can ask your questions. So now, do you have any doubt or any questions?
I think,
Any
sir,
participant? Yeah, Madhur ma'am.
ma'am, there's so much of information. It was like we never know all this. We just know that we can write a program and we think of input, but there's so much of things happening. Like when sir was explaining, it was like, oh God, there's so much that we don't know. We just look at data and we write a program, we classify, but there's so much of, sir, in bio processing, like you deal with so much of data and so much of real stuff, the real processes,
Yes.
the way the cancers. Yes, sir, it is so amazing. And to know really, we write a program and we think of the data and how to classify, detect, but then at the end of the day, it's the cells, and you are talking about the cells. It was like
Yeah.
everything was like, oh God, is it was uh, like we don't know in such depth. You have explained how the cancer cells, how they release the material which can be detected, and how with the techniques that we with machine learning AI, how the time is being the time of predicting, detecting, how it is actually 15 minutes the slide, and there you were saying it is like so 1 to 3 hours and 15 minutes. So with so many patients, like in places like in India especially, where there's so much of population and it takes days to get reports. So this
Yeah.
is like very like very appealing, like.
Yeah, actually, yeah, there are many areas where like biomedical or this machine learning can be integrated. Like I just gave a certain example because I have been working particularly on glioma and this neurodegenerative disease. So there, so far there is no drug which can
Sure.
cure this. Both diseases are like fatal, like this Alzheimer disease. Once it happens, like dementia will start, and then there will be a then there will be a onset of neurodegeneration, and the person will start forgetting everything, and it will be a a very dramatic havoc for all uh, the relatives even. And similarly, like the glioma, it's also known as death on diagnosis. So within 1 year or 1.5 years, the patient will die once this glioma occurs, if it is in certain if it is not not in the periphery, so even right after diagnosis, even for the even for the operation, it costs huge amount of money, and also it's a burden. And for example, even if you want to cure, even if you want to diagnose this, you have to take some certain tissues from the brain, and then you have to do the various you have to run various kind of experimental methods, like immunohistochemistry, or even you have to do HNE staining. The doctor will tell you to do the biopsy, tissue biopsy, and in that one there is a need for the tissue extraction, and it is again an invasive process, uh, which cause a lot of pain even. It's not like that easy to open the brain and get the sample. So here comes this machine learning in the picture. Like if we have certain methods, like some deep learning based methods, where we can train our model systems, like what are the features which are easily available in our blood, or even the cerebrospinal fluid through the spinal cord, so that might facilitate, like make this whole process smooth, but yeah, I know this is not that easy, but there are advancements in this field, and still
So,
ongoing. Yeah. Yeah.
so you were also talking about biosensors, like these sensors are injected, and that's how the data is like received, and then the machine learning part, like.
Yes, yes. So biosensor, why I talked about is because this is one of the advanced tool in the hospital, like even like surface plasmon resonance. In most of the hospital, this is being used for the diagnosis, like within 30 minutes or 1 hour. Uh, the technical staff can send the report to the doctor, and you will get to know whether you have a cancer or not, like by even there are like flow cytometry based biosensors, so these are already being used in the as a
So
biochemical
these
testing
techniques
in
are
most.
available in India? Like, are these techniques
Yes, of
available
course,
in?
of course. These biosensors are also available in India. Yeah. So
So
these
the
are all
students.
these are used for the diagnosis. So, but to reduce this time consuming methods, or even to reduce the cost of this, so in that case, we can propose some machine learning based applications.
Okay. Sir, a student here has a question in the chat box. He's saying in the beginning when you told us about the neurons that destroy the power of thinking, like you gave an example of Stephen Hawking. Does it affect the brain only? Is there some way to cure it?
Or actually, Stephen Hawking case, yeah, it's a famous case, so that's why I just gave it as one of the example. So Stephen Hawking was suffering from ALS, amyotrophic lateral sclerosis. So that is not exactly Alzheimer, but that has a similar kind of pattern, that is neurodegeneration. So you are asking about how do you know that destroys our power of thinking? So not power of thinking, but power of remembering. That is dementia. Dementia. So that, yeah, that happens in most of the neurodegenerations, including what I talked about is Alzheimer. So of course there are certain cure, but there is no complete cure, or like once there is onset, yeah, the person will start showing of dementia. They will he or she will start forgetting things, and the life will become miserable, and yeah, of course there is no exact cure. That's why the NIH highest grant is for the neurodegenerations, like neuroscience. Most of the brain diseases, they get highest amount of money for doing the research. Yeah. So yeah, there is
That
no exact cure.
so, Ashtik, that answers your question. Sir, I have another query, sir. Like,
Yes, yeah.
since we're talking
Go
about
ahead.
Stephen Hawking, there's another personality whose complete health was always on the internet. Michael Jackson. So it was not actually a kind of cancer, but then he had uh, it's also said that he had a kind of disease in which his skin was whitening. So is it a kind of cancer? Like somewhere I read it is again a rare disease that he had. If you have any
No,
information.
I think he was suffering from vitiligo. So that's not a cancer, so
Okay.
that's not a cancer. And also, like but it is not treatable, it cannot be treated, although the many doctors will claim, but there is no exact treatment for that. But of course, people, like, it depends on how far it has gone. Like if it is universalis, vitiligo universalis, then whole body will become white, because it's an autoimmune disease, and there is a production of antibody for itself, and it starts eating their own cells, and that's, yeah, there is a whole mechanism for this vitiligo, but there is no exact cure for that. So Michael Jackson was suffering from vitiligo, so that's how, because he was an African guy, African American guy, and he was like suffering from vitiligo, I would say, yeah. So, and he suffered from, I think, universalis. So that's how it progressively grown up to completely white, from black to white. So many people claim in their videos or YouTube videos that he had this disease or that disease, which was like cancer or something, blah blah, or he changed his skin color. But basically he was suffering from vitiligo, and of course there was like, he did some skin treatment, but those might be in vain, I think, yes, and that's why he changed his whole body color. Yeah, but we haven't seen his whole body, so we don't know. But of course, we know that his face was changed, so he did some plastic surgery. Yeah,
Yes.
but vitiligo, that disease was vitiligo.
So, anybody,
Okay,
Vishnu ma'am, that's why I
yeah,
can
yeah.
ask so
Actually,
many
I want,
questions.
yeah, I actually want to, actually, I wanted to ask you how this machine learning can help in diagnosis of the brain cancer, but you have already explained clearly, so now I got the idea. But the thing is that how it can be helpful, whether it can be helpful in diagnosis of coronaviruses, how it
Yes,
can be
yes, of course.
helpful, machine learning.
Actually, yeah, many people have been working on the COVID-19. So there are there are a lot of machine learning related paper. Even I have been working on the COVID-19
Yeah, so
with
that,
certain
yeah, I
diseases.
saw your
And
paper that means you are doing work. Yeah, you were working on this, so that's actually I'm asking.
yes, yes. So if you directly search in 2021, there is
Yeah.
already, okay, I'll show you one example. Can you see my entire window?
No, till now we are not able to see. Your
I
screen is
see.
not shared actually.
Uh, a window tab. Yeah, can you
So
see
there are
this?
three
My can you
dots
see
in.
this? Can you see this interface?
No, yeah, yeah, now it is coming.
Can you see this one? This paper.
Yeah, 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 yeah. Yeah, now it's
So
coming.
see,
Yeah, the paper
so
is
see
there.
in this nature publication journal Digital Medicine, machine learning based prediction of COVID-19 diagnosis based on symptoms. So people have been using, and you can see like what kind of data they have used. So you can easily read this kind of paper. So people have been using. So there are so many papers people are using, like COVID-19 projections using machine learning. Yeah. So
So one of the
even I also published one, like it's still under review in the nature paper. You can search. So this paper is still in the preprint. So we model the COVID-19 with human pluripotent stem cell. So we modeled this whole system using this. Yeah, you can go and read this kind of stuff. Yeah, no problem. I think you'll have a good picture of it. Yes.
Okay. So now,
in the
any questions from audience side? I mean, from participant. So you can see now your machine learning can be used in anywhere. It can be used in, you can say in diagnosis here, brain cancer. Not only in brain cancer, you can use in anywhere, everywhere in the COVID-19 also. Dr. Thakur is also using machine learning concept. So, do you have any questions? Yeah, otherwise, I will ask Madhuri ma'am to give the vote of thanks to Dr. Abhimanyu Thakur.
Welcome. Yeah, sure.
Sir, I have a
Madhuri ma'am,
question,
please.
sir. Sir,
Yeah,
I
sure.
have a
Yeah, go ahead.
question before. Sir, in your presentation, you have shown us a slide where you essentially have used support vector machine with 4 or 5 different kernels. So, in this presentation for diagnosis of cancer, like,
Hello.
yes, sir.
Yeah, yeah. What's your question then? Yeah.
Yes, sir. Sir, so which was the best technique out of like? So it depends on various other features of what we are taking the cells. Like,
Actually, there is no best or something. I mean, in my view, but I think so far in what we chose among them, we found like Bactri worked better. So I think it all depends on training data set, and also uh, like what are the how many different features you are providing to the that machine learning model.
sir.
So it depends on scenario, yeah, and also like what kind of algorithm you use for that.
Yes, sir. Thank you, sir. Sir, I actually would like to thank you very much because this is 6 a.m. I think at your side, and you are giving us time at morning 6 a.m.
It's
when we
fine.
get up. And thank you so much for taking all the effort to tell us and take us through this journey, especially the biology part, the phenomenon, the mechanism of how things work. We never get into it, and even if we read, we could not understand the way you have presented and explained to us. Thank you so much for your time, sir. I also want to thank the participants. I know since morning you had classes. Some are also somewhere texting me that sir, data pack and things like that also have a challenge. Due to COVID, we were having online classes, but those of you who could join, thank you, and I hope you did learn and did get an experience of what machine learning can be done in different areas. Like today, we sir has explained us how it can be used for machine for cancer diagnosis and research, and at the same time he also showed us how it is being used for COVID, which is COVID-19 diagnosis. So this has been very useful session for us. Thank you so much, sir. Thank you, Vishnu Priya ma'am, and thank you to my HOD ma'am. Though she is not here because she was busy with the conference, Sarda sir, Divya Sundar sir for live streaming the session, and all the participants. Thank you, everyone. Thank you, sir.
Yeah, thank you. Yeah, bye. Take care.